So in this experiment, we are going to look at the anaerobic jar. And so this will be day 1, the setup. And this is activity 2-8. So the purpose of the anaerobic jar is to determine the oxygen requirements of an organism. Meaning, do they grow when only oxygen is present? Do they grow when only oxygen is absent, meaning anaerobic? Or do they grow both aerobically and anaerobically, meaning they can grow with or without oxygen? And so to do this experiment, you would do this in pairs, and per pair, you would get 2 nutrient agar plates. One plate you would label the aerobic plate, and the other plate you would label anaerobic. And so what you would do is you would divide your plate into thirds, and you would label one quadrant PA for Pseudomonas aeruginosa. You would label one quadrant Staph aureus, so SA, and then the third quadrant you would label Clostridium sporogenes, CS. And so you would do that on both plates. You want to have those 3 organisms on both plates. So to do this, all you would do is you would inoculate this just like you would for the starch hydrolysis or the casein hydrolysis, where you would use your aseptic technique. You would flame your loop, pick up the broth, so you would get Pseudomonas aeruginosa, the bacteria, in a broth using my loop, and then one broad streak. And then you would repeat that for each organism. So you would end up with 1 streak for Pseudomonas aeruginosa, 1 streak for Staph aureus. Clostridium sporogenes, sometimes we draw 2 lines. We do 2 broad streaks just because it might not grow as well. And so sometimes we will inoculate 2. If you only did 1 broad streak, that would probably be fine. But sometimes we tell students just to be safe to do it twice, just so you can see the growth. So you would have your 2 plates that are inoculated in duplicate. 1 plate would be your aerobic. This plate is the plate that would go in the incubator. In our incubator, oxygen would be present, and so that would be our aerobic plate. Our anaerobic plate, on the other hand, has to be without oxygen. So we can't just take that plate and put it in the incubator, because the incubator is going to have oxygen. So in order to make the other plate anaerobic, we have to take that plate and we need to put it in a setup that is called an anaerobic jar. And so I'm going to describe how our anaerobic jar works. So here is our anaerobic jar. It is basically a chamber, and within that chamber we have these chemicals. And these chemicals are there to make our jar anaerobic. So let me explain how this would work. So what we need to talk about first is we need to have an understanding of what is called redox reactions. And a redox reaction is going to be an oxidation and a reduction. And so for me, the way that I remember what is an oxidation and what is a reduction is, for me, I remember oil rig. And that is the O. Oxidation is a loss of electrons. So notice OL. So oxidation is a loss of electrons. And similarly, you can follow the hydrogens. If something loses hydrogen, it's losing the electron that goes with it. So oxidation is going to be a loss of electron. A reduction is going to be a gain, right? So reduction is a gain of an electron. Similarly, a gain of hydrogens. So if something gains hydrogen, it becomes reduced. Now, in terms of these redox reactions, those are always going to be coupled. You're always going to have an oxidation paired with a reduction, because something has to lose the electron and something else has to gain it. We're simply relocating electrons from one place to another. So in the case of our anaerobic jar, so here's our anaerobic jar, the way that this works is we need to remove oxygen. And so to do that, we have these chemicals. They're in this little pack here. And these chemicals are referred to as anergen chemicals. And these anergen chemicals, their job is to reduce oxygen to form water. So notice that this oxygen is going to become reduced. It's going to gain electrons, it gains hydrogens, and that's why it becomes water. So these chemicals, their job is to reduce the oxygen to form water. Now, when it does that and it's in this sealed chamber, what that does is any O2 that was present is now reduced to form water. And now our jar is anaerobic, right? There's no more oxygen in the jar. And so the condition in that jar would now be anaerobic. And so these anaerogenic chemicals are there to make that jar anaerobic. And that is because they're able to take the oxygen and reduce it to form water. And so what you see when you actually do this chamber, you'll often see condensation forming inside the jar. And that condensation is, again, the result of the oxygen being reduced to water. Now, when we set this up, we need to make sure that, in fact, our jar is anaerobic. We need to make sure that our seal is good and that, in fact, those chemicals are reducing the oxygen to form water. And so the way that we make sure that this is working is that we also put in the jar an O2 indicator. And this O2 indicator is called resazurin. So resazurin is going to be pink when oxidized. Oxidized, think of with oxygen. That's your aerobic. So if I take that resazurin strip, it's a little piece of paper. If I take this little strip and I leave it out on the desk, that strip is going to turn pink because the oxygen in the atmosphere is going to interact with the resazurin and it's going to turn pink. The resazurin is going to be clear when it's reduced, meaning when it's anaerobic. And so when I put my indicator, my resazurin, in my jar, initially it's going to start out pink because it's exposed to the oxygen. However, once I put my anaerogen chemicals in there with my plates and then I seal it, if in fact this chamber becomes anaerobic, meaning if my anaerogen chemicals work and they reduce the O2 to water, what's going to happen is that the resazurin strip is going to go from pink, meaning O2 is present, and it's going to become colorless. And so one of the things when we set up our anaerobic jar, we're going to notice that the resazurin is pink to start with. However, once we take this jar, once our jar is set up, and we take that jar and we put it in the incubator, when we check those plates at 48 hours, one of the things we want to look at is we want to look at that resazurin strip. And we want to make sure that, in fact, when we look at our jar after 48 hours, that strip is going to be colorless or clear, meaning it's just going to be the white paper. Because we need to ensure that the jar is, in fact, anaerobic. If it's not, we can't make a conclusion as to whether bacteria grow aerobically or anaerobically. And so we can't make that conclusion if the jar is not, in fact, anaerobic. And we know that the jar is anaerobic because of the resazurin strip. You want to see that resazurin strip being clear after the 48 hours. That would indicate that the jar is anaerobic. So what I have down below is a link for a video, and I will include this on our page, on our Canvas page right underneath this video. And basically it's just going to demonstrate and show you how we would put this jar together, just so you can see. It's not my video, so I don't want to include it within my PowerPoint, but I will link it in Canvas so that you can watch that to see how this is actually set up as well. So now let's look at our anaerobic jar day 2. Looking at our readout. So meaning once we take our jar out of our incubator, let's talk about what the results would mean. So again, just to remind you, for our anaerobic jar in terms of our setup, we have this canister, and within it we have these anaerogen chemicals. And these anaerogen chemicals, again, their purpose is to reduce O2 to water. Basically to make the jar anaerobic, to make the jar so that O2 is no longer present. We would also have our anaerobic indicator or our O2 indicator, In our case, it would be our resazurin. And again, when we look at our jar, we would need to see that to be clear, or the white paper, meaning that O2 is not present. If that strip, when we take our jar out, if that strip is pink, that tells us that oxygen is still present in that canister, and that canister was not in fact anaerobic. So our resazurin has to be clear when we look at it. And then we had our Petri plates that are within the jar. And so after the 48 hours, after we grow these plates, we would unscrew the lid. So we would unscrew the lid, open the jar, and we would take out our plates and look at to see, do we see growth on the anaerobic plates? Do we see growth on the aerobic plates? Remember that the aerobic plates would be the ones that were in the incubator. So for each of our organisms, we grew them aerobically in the incubator and anaerobically in the anaerobic jar. And so we want to see, where does bacteria grow? Does it grow only aerobically? Does it grow only anaerobically? Or does it grow under both conditions? And so the next set of slides are basically just kind of explaining about different O2 requirements. These slides are a little bit different. The ones you'll see in this video are a little bit different than what's in your lab PowerPoint. The slides are actually pulled from chapter 6 from lecture. So same content, the slides might look a little bit different, but the idea is the same. And it's talking about basically the O2 requirements for different organisms. And I talk about in that about how organisms that live in oxygen have to have specific enzymes to be able to detoxify what we call reactive oxygen species, meaning oxygen forms of oxygen that are unstable and highly reactive. And so organisms that grow where O2 is found need to have those enzymes to basically neutralize those reactive oxygen species, to make it so that they're not toxic to the bacteria. And so I'm going to walk you through and talk about what those enzymes are there for, what processes do they neutralize, and then also what, as a result, would be an organism's oxygen requirements. And so we're going to go through some background information about O2 requirements for cells, and then we're going to talk about what were our results in our anaerobic jar.
Organisms that grow where oxygen is found have developed enzymes that scavenge and neutralize reactive oxygen byproducts. And so this is going to be a two-step process, and this two-step process requires two enzymes. The first enzyme is called superoxide dismutase, which you will see abbreviated SOD. And this enzyme is used to neutralize superoxide ions. So they will neutralize those superoxide ions, and they will convert those superoxide ions into our products. And we have two products in this case. We have hydrogen peroxide and we have O2 gas. Now, remember I said that hydrogen peroxide is also unstable. So there's going to be a second enzyme needed to neutralize the hydrogen peroxide. But first, let's talk about what types of organisms would have SOD. And so this is used by organisms that grow in the presence of O2. So whether or not the organism uses oxygen doesn't really matter. Organisms that grow in the presence of oxygen need to have that superoxide dismutase. They need to be able to neutralize those superoxide ions that can form. Now, when superoxide ions are neutralized and they produce hydrogen peroxide, we need a second enzyme to neutralize the hydrogen peroxide. And so that second enzyme is one that we talked about in lab, and that enzyme is catalase. And so this is used to neutralize hydrogen peroxide. So catalase is an enzyme that's used to neutralize hydrogen peroxide. So it's going to take that hydrogen peroxide and it's going to neutralize it, and the products are going to be water and O2 gas. So notice that in this case, we form more O2 gas. So for catalase, it's not just used by organisms that grow in the presence of oxygen. In this case, it's actually used by organisms that use O2 as their final electron acceptor. So not only do they grow in the presence of oxygen, but they actually use O2 as their final electron acceptor. Because if the organism does not use O2, they're not going to want to use catalase and produce more oxygen. Notice that this product is going to produce more O2. That doesn't want to happen if the organism is not going to utilize that O2. So some organisms have a third enzyme instead of catalase. They have an enzyme called peroxidase. And so for peroxidase, we still have hydrogen peroxide being neutralized, but our products in this case are going to be simply water. It's not going to form more O2 gas. Now, I didn't balance that equation. I'm not worried about you knowing that, but I want you to know what the purpose of those enzymes is. So peroxidase is used to neutralize hydrogen peroxide, and it's going to produce water in the process. It's not going to produce more oxygen. So this is used by organisms that grow in O2, but don't use it. And again, that's because if the organism does not use O2, it doesn't want to form more O2 gas. So instead of using catalase, instead the bacteria would use peroxidase. They would still neutralize hydrogen peroxide, but they're going to form the product of just water. And so again, it's the presence of that catalase that's going to cause the bubbles when you pour hydrogen peroxide in a wound. The hydrogen peroxide that causes the bubbles is not only from bacteria that might have gotten into the wound, but your cells also contain catalase. And when you get a cut and those cells are damaged, those cells will also release catalase, which would also then produce the oxygen bubbles when you pour hydrogen peroxide in that wound. And so these are the enzymes that bacteria need to have if they're going to grow where O2 is found. They have to have these enzymes to be able to neutralize those toxic oxygen species. So we have different terms for an organism's oxygen requirements. If we call the organism an aerobe, what that means is that the organism uses molecular oxygen, so O2, as the final electron acceptor. So as our final electron acceptor in aerobic respiration. And so within the umbrella of aerobes, we can have organisms that are obligate aerobes. And just like the name suggests, they are obligated or they require O2. And an example of this would be Pseudomonas aeruginosa. And Pseudomonas aeruginosa causes infections in burn victims and patients with cystic fibrosis, which we will abbreviate CF. So Pseudomonas aeruginosa causes infections in burn victims, and in fact, it's the leading cause of death in burn victims. And you might recall that Pseudomonas also produces a pigment called pyocyanin. Pyo again refers to pus, cyan refers to blue, which means that when this organism is growing in a wound, it causes the wound to appear to have blue pus. And so that is a hallmark of a Pseudomonas aeruginosa infection. It also is very problematic in patients with cystic fibrosis. Cystic fibrosis, you may recall, is a genetic condition that people are born with. And in cystic fibrosis, patients have defective chloride channels. Because they don't move chloride properly, they also have problems with water imbalance, and they end up with these secretions in the lungs. And Pseudomonas really likes water. And so if Pseudomonas happens to get in these secretions in the lungs of these patients, it causes a major problem because they can't clear it out of their lungs. And so Pseudomonas aeruginosa would be an example of an obligate aerobe. Other examples of obligate aerobes would include most fungi, protozoans, and many other bacteria such as Bacillus species. So you might recall that Bacillus is a bacterium that produces endospores, and it is an aerobic species, meaning that it grows where O2 is required. And also under obligate aerobe, we have Mycobacterium tuberculosis, which causes lung TB, tuberculosis. Next, we have our microaerophile. Micro refers to little, aero refers to air, phile is lever. So these are our little air levers. These require low O2 concentrations. And so an example of this would be Helicobacter pylori. And you might recall that Helicobacter pylori is the infectious cause of ulcers. And so what happens is that if Helicobacter pylori, if this bacteria happens to get into the stomach, remember that an advantage that Helicobacter pylori has is that it produces urease enzyme. And what that does essentially for this bacteria is that if this bacteria gets into the stomach, in the stomach is the substrate for the enzyme, urea. And so if Helicobacter pylori gets into the stomach, it's going to take that urea in the stomach and it's going to hydrolyze it. It's going to break it down. And one of the products that gets produced during this is going to be ammonia. And the advantage for the bacteria to produce ammonia is that the stomach is a very acidic environment. Most bacteria don't survive well at that acidic of a pH. So because Helicobacter pylori produces urease, it's able to produce ammonia, it's able to neutralize the stomach acid, and therefore grow in the lining of the stomach, and it erodes and it damages the lining of the stomach, which then leads to ulcers. And so this bacteria is the infectious cause of ulcers. And again, remember that not all ulcers are caused by bacteria. Other examples of organisms that are microaerophiles would be organisms that live in soil, in water, or in mammalian hosts in places that are not directly exposed to the atmosphere. So another example of a bacteria that would be a microaerophile would be something like Neisseria gonorrhoeae. Neisseria gonorrhoeae, remember, is the cause of gonorrhea. Gonorrhea is a sexually transmitted infection. And this bacteria is a microaerophile. It needs a low concentration of oxygen. And so this bacteria is going to be found on the opening of the urethra. So it's not in full contact with oxygen. It's not fully anaerobic, but it has a very low concentration. And so that would be an example of a microaerophile. If we call an organism an anaerobe, what that means is one of two things. Either the bacteria does not require O2, so in some cases they use O2 if it's available, but they can grow in the absence of O2. In some cases that means that they don't use O2 at all, or it could be that O2 is actually toxic to them. They're actually missing those enzymes that we talked about in the last slide, which allows the organism to be able to detoxify those reactive oxygen species. And so we're going to look at some different examples of some anaerobes. So the first one that we're going to talk about is going to be the obligate anaerobe. And for our obligate anaerobe, it's going to cease growth or die in O2. So cease growth or die in O2. Notice I did not just write that it causes them to die. And the reason for that is that an example of an obligate anaerobe in many cases is the genus Clostridium. So many Clostridium would fall under being an obligate anaerobe. Now, if you recall, Clostridium produces a structure when conditions become harsh or unfavorable. Clostridium would produce the structure to protect themselves. Now, in the case of Clostridium, that condition could be that oxygen is present. So if O2 is present and O2 is toxic to them, they would produce the structure that would protect them until conditions became favorable again. And so think back to what we've learned and think about what structure Clostridium produces that allows them to not die from O2, but instead would allow them to cease growth. And so what structure would that be? And so if you were thinking endospores, you are correct, right? Because endospores are a structure made of keratin that bacteria can produce when conditions become harsh. In the case of Clostridium, because O2 is toxic to them, in the presence of O2, they would form this endospore to wait it out until conditions became favorable again. And so that's why I was careful to write cease growth or die in O2, because in some cases it's not necessarily going to kill them. However, it will at least inhibit their growth. So we're going to put example, Clostridium perfringens. And Clostridium perfringens causes gas gangrene. And so you might recall that we've talked about this organism, and that is that if Clostridium perfringens happens to get into a wound, it can start to cause necrosis of the tissue. The tissue starts to die. And as a result, when the tissue dies, that makes the environment anaerobic. Because if the tissue is dead, it's no longer getting blood flow. And if it's not getting blood flow, it's not getting enough oxygen. And that further helps Clostridium to be able to grow. And so what happens is that tissue that's necrotic, that has died, the bacteria is going to start to grow and to spread, and that tissue is going to continue to die. And remember that we talked about that oftentimes, because it's difficult to get the antibiotic to the site of the infection, one of the more common things that happens in response to gangrene is that oftentimes the treatment involves amputation of the limb. So if it was the finger, it might be the finger. If it was the whole hand, it could be the hand. But basically that the treatment might include amputation because it's difficult to treat gangrene. Now, there have been some examples of using hyperbaric chambers. So basically to put the patient in a high oxygen environment, which remember that Clostridium does not grow well in oxygen. It inhibits their growth. And so this would help to hopefully treat Clostridium. However, in severe cases, sometimes that's not enough. The hyperbaric chamber is not enough, and they still end up having to do an amputation anyways. And so this would be an example of an obligate anaerobe. Other bacteria that would be obligate anaerobes include often intestinal bacteria. So bacteria that live in the intestinal tract would often be an obligate anaerobe. For them, oxygen is toxic. The next category of organisms that we have would be our facultative anaerobes. And our facultative anaerobes grow with or without O2, but grow better. They grow better with O2, meaning that they will use it when given the choice. So for these organisms, they do use oxygen. However, they don't require it. And so that's why I wrote grows with or without oxygen, but grow better with. Now, the reason that they grow better with oxygen is that if the organism were to do aerobic respiration using O2 as the final electron acceptor, if they use oxygen as the final electron acceptor, they can get 30 to 32 ATP per glucose. On the flip side, if the organism grows anaerobically and they use fermentation, they only get 2 ATP per glucose. So notice that using aerobic respiration gives you a much higher ATP yield than fermentation does. And so if the organism is a facultative anaerobe, right, they can grow with or without. But if given the choice, they're going to grow with O2 and use O2 because for them, using oxygen gives them the highest ATP yield. And so an example of this that you've seen this would be when we did our standard plate count. So remember that our standard plate count, the purpose of our standard plate count was to isolate individual colonies of bacteria and determine the titer, so the concentration of bacteria in the solution. And so when we did this, we took our E. coli and we mixed it with the agar and then poured our plates. And so you might recall that we had some colonies on the top of the agar and some colonies embedded in the agar. And if you recall what that looked like, you might recall that the colonies on the top of the agar were very large. And the colonies that were embedded in the agar, that were in the middle of the agar, those colonies were teeny tiny little dots. And again, back then I told you that the reason for that is that if O2 is available, so the top of the agar, bacteria is going to get a lot more ATP per glucose than the bacteria that's growing anaerobically within the agar. And so that allowed you to see that in fact facultative anaerobes grow better with O2. So an example of this, E. coli. So Escherichia coli. Okay, you can write out Escherichia, you've seen it many times. So example is E. coli, and E. coli, for example, is the cause of urinary tract infections. About 90% of all urinary tract infections, what we call UTIs, are caused by E. coli. And so E. coli would be an example of a facultative anaerobe. In lab, we also saw another example of this, like Staph aureus, for example. Staph aureus is also a facultative anaerobe. And many gram-negative intestinal bacteria would also fall under being a facultative anaerobe. So those would be examples of facultative anaerobes. And the last one is going to be our aerotolerant anaerobes. Aerotolerant means that they tolerate O2, but they don't use it. So we're going to write can grow in O2, but don't use it. So we can put can grow with or without oxygen. So notice there's a key distinction between a facultative anaerobe and an aerotolerant. Both of those two categories can grow with or without oxygen. The difference between them, however, is that the facultative anaerobe will use O2 if O2 is present. An aerotolerant anaerobe can grow with or without, but even if O2 is present, it's not going to use it. It's not going to use it as its final electron acceptors. For example, examples of aerotolerant anaerobes would be Streptococcus, Pyogenes, which causes strep throat, and these organisms are strict fermenters. So for strep pyogenes, even if O2 is present, it's not going to matter. It's not going to use O2 even if O2 is present. Other types of bacteria that would fall under being aerotolerant anaerobes, some lactobacillus, for example, other streptococcus species, etc. So those would be examples of aerotolerant. They grow with or without O2, but they don't use it. And so again, you want to be careful. That's the distinction between the facultative anaerobes and the aerotolerant, is that the facultative anaerobes can grow in both conditions, but if O2 is present, they will use it. Aerotolerant is not going to use it. So now let's talk about why organisms are one of these categories of organisms. And so you might recall that in lab, we talked about our thioglycolate broths. And that our thioglycolate broths use a reducing media to create an anaerobic environment. And so in lab, we talked about what was in thioglycolate broths, and you might recall that there are two chemicals that are in the thioglycolate broth, which helps to make the environment anaerobic. And so if you recall back, we talked about L-cysteine, which is a dimer of cysteine amino acids. So we had L-cysteine and we had our sodium thioglycolate. Those were our ingredients that both were used to reduce O2 to water, which makes the bottom of that tube anaerobic. You might also recall that in this media, we have an O2 indicator, and that O2 indicator was rizagirin. And rizagirin is pink when O2 is present. And so we talked about what our thio broths look like for each type of organism. So again, if it's an obligate aerobe, it's going to grow only in the top, only where the rizagirin is pink. In the facultative anaerobe, it's going to grow best where O2 is present, so best where it's pink, but it will still grow in the bottom part. It'll still grow in the anaerobic part. In the case of an obligate anaerobe, it's not going to grow where the tube is pink. It's going to grow towards the bottom. It might be settled at the very bottom. It might be all throughout the part underneath the pink. But the idea here being that it's not going to grow where it's pink, because pink is where the O2 is present. Next, we have our aerotolerant. Aerotolerant, remember, means that they tolerate O2, but they don't use it. So what you're going to see in this example is that it might grow evenly throughout. It grows equally well in the top as it does in the bottom. And then lastly, we have our microaerophiles, and our little air lovers are going to grow right on the junction between where it's pink and it's not pink. And so in lab, we saw it growing right underneath where the pink is. So now, what I want you to think about is, for each of these types of O2 requirements, I want you to think about whether or not they would have superoxide dismutase. So meaning give it a plus if they produce it or a minus if they don't. And same thing with catalase. Does the organism produce catalase? If it does, give it a plus. If it does not, give it a minus. And so take a minute, and I want you to attempt this on your own before hearing the answer. So if you need to, pause your video, and when you're ready, turn it back on, and I'll go over the answer with you. Okay, so hopefully you tried this on your own, but let's review. So superoxide dismutase, remember, is used to neutralize superoxide ions, so superoxide radicals. So for our superoxide dismutase, remember that it's produced by organisms that grow in O2. So whether or not they use O2 does not matter. If they grow where oxygen is present, they will produce superoxide dismutase. So obligate aerobes, do they produce superoxide dismutase? And the answer is yes, right? They grow where oxygen is found, therefore they must have SOD. Facultative anaerobes grow with and without because they grow where O2 is present. They will have their superoxide dismutase. Obligate anaerobes do not grow where O2 is found. They do not have superoxide dismutase. And so in this case, the organism is an obligate anaerobe. What makes them an obligate anaerobe is the fact that they don't produce this enzyme. They don't have this gene. They don't produce this enzyme. And as a result, because they can't neutralize superoxide ions, those superoxide ions would be toxic to them and would kill them. And that is why they are an obligate anaerobe, because they do not produce that superoxide dismutase. So that would be negative. Aerotolerant anaerobes do grow where O2 is found. That would be positive. And then lastly, we have our microaerophile, our little air lovers. They do grow where O2 is found, but only where we have a low concentration. And that's because they do produce SOD, but only a little bit. So they have just a little bit of SOD present, which is why they can grow where O2 is present, and they actually even use O2. But they can't grow where oxygen concentrations are high because too much of those reactive oxygen species would be toxic. They don't produce enough enzymes to be able to neutralize the full amount of oxygen in the atmosphere. Next, when we look at catalase, catalase is not produced by organisms that grow in O2, but instead is produced by organisms that use O2. So that's our distinction. So now when we're talking about catalase, catalase is neutralizing hydrogen peroxide, and it's produced by organisms that use O2 as their final electron acceptor, not just grow in the presence, but actually use it. So if we look at our obligate aerobes, would they produce catalase? The answer is yes, because they use O2. Facultative anaerobes, they still use O2 as their final electron acceptor. Therefore, they would produce catalase as well. Obligate anaerobes, right? They don't use O2. They are catalase negative. So again, the reason that obligate anaerobes are obligate anaerobes is because they're negative for SOD and catalase. They don't produce either of those enzymes, and therefore O2 is toxic. Now, when we get to our aerotolerant anaerobes, remember that those grow where O2 is found, but they don't use it. So this organism is going to be negative for catalase, but remember it's going to have peroxidase instead. And you might recall that peroxidase is used to neutralize hydrogen peroxide, but it doesn't produce more O2 as a result. It just produces water. So aerotolerant anaerobes will not produce catalase because they don't use O2. And then our microaerophiles, these would produce a little bit of catalase. And again, that's what allows them to live where some O2 is present, but only in low concentrations.
So here are results for the anaerobic jar experiment. On the left, we have our aerobic plate, meaning this was the plate that was grown with O2 present. This is the plate that was grown in the incubator. And on the right, we have the anaerobic plate that was grown in our anaerobic jar. And so if I look, PA, Pseudomonas aeruginosa, notice that it grew on the aerobic, but it did not grow on the anaerobic. And I'll go over these terms with you in a minute. Notice that Staph aureus grew both aerobically as well as anaerobically. If we look at Clostridium sporogenes, did not grow aerobically. It's not there aerobically, but it did grow anaerobically. So Pseudomonas grew only aerobically, Clostridium grew only anaerobically, and Staph aureus grew under both conditions. And so let's talk about what those terms are. So before we talk about those terms, there's another part to this experiment that I'm going to include in this video rather than a separate video, and that is the thioglycolate media. And so the purpose of this experiment is to determine the O2 requirements of an organism. So similarly to what we saw for the anaerobic jar, but this actually gives us a little bit more information. And so let's talk about what is in our thioglycolate broth. So first ingredient, yeast extract and peptones. Those are there, like we've always seen, as food for general growth. Those are just food for general growth. They're there to help support bacterial growth. Next, we have glucose or dextrose, which is there for fermentation. It's just sugar to help bacteria grow. The next two ingredients both have the same function. And the function of the sodium thioglycolate and the L-cysteine is that their job is to reduce O2 into water. So just like in the anaerobic jar, how we had our anagen chemicals, and those chemicals' job were to reduce O2 into water, same idea in our thio broth. Our thio broth is going to have these two ingredients. It's going to have sodium thioglycolate and the L-cysteine to help reduce O2 into water, again, to make it anaerobic. Now, the L-cysteine is basically a dimer of cysteine amino acid, meaning that it's these two cysteine amino acids that are linked together by their R group, which is linked by what's called a sulfhydryl bond. And that is going to allow it to help reduce O2 into water. And so we have two chemicals in there that their job is to make it anaerobic. So because we want to have a portion of our tube that is anaerobic and we want a portion of our tube that is aerobic, so we can see under which condition do bacteria grow. Now, how do we know which part of the tube is aerobic versus which part is anaerobic? Well, just like in our anaerobic jar, how we used Resazurin as our O2 indicator, we are also going to do that in the thioglycolate media. And so in our thioglycolate media, the Resazurin is going to be our O2 indicator. And remember that it's pink when oxidized, meaning O2 is present. So notice that if I look at my thio broth, this is an uninoculated thio broth. At the top it's going to be pink. And it's pink because that's where O2 is present. However, when we go further down in the tube, these two chemicals are going to be making the tube anaerobic. And so this portion of the tube is anaerobic, and the residuant is colorless, meaning we don't see a color because the residuant is reduced and O2 is absent. And so where it's colorless, this is where the tube is anaerobic. Where it's pink is where the tube is aerobic. And so this allows us to see where do the bacteria grow. So let's go through and fill in this table. So our first organism is going to be our Pseudomonas aeruginosa. And Pseudomonas aeruginosa is a gram-negative rod. That's what this abbreviation stands for. This is a gram-negative rod. And did it grow on our aerobic plate? And the answer is yes. It grew on the aerobic plate. Did it grow on the anaerobic? Answer is no. Did not grow on the anaerobic plate. So it grew aerobically, but not anaerobically. So we call this their O2 requirement. This organism would be referred to as a strict or obligate aerobe. So this organism is a strict obligate aerobe. It only grows aerobically. Now, what diseases is this associated with? Well, there's a variety of diseases that this is associated with. Pseudomonas aeruginosa is a major cause of death in skin infections in burn victims. It also can cause ear infections. It can cause a lung infection in cystic fibrosis patients. Patients who have cystic fibrosis, they have a genetic condition in which they have defective chloride channels. And as a result of having these defective chloride channels, they don't move water properly. And so these patients end up with this thick secretion in the lungs, and it's more susceptible to these Pseudomonas aeruginosa infections. And so that's why often cystic fibrosis patients are advised not to date other cystic fibrosis patients because the chance of transmitting Pseudomonas between each other would be high, and they're very susceptible to that infection. That type of infection can be fatal for them. And so Pseudomonas is an organism that really likes water. And so notice ear infection, when you get water trapped in your ear, in a burn, it's really moist, right? And if the bacteria gets trapped under the bandage, it can cause an infection. Now, one of the interesting things about it is it produces this pigment called pyocyanin. Pyo refers to pus. Cyan refers to blue. So this produces this blue pus pigment. And so when it gets into these burns and it grows, well, Pseudomonas can produce that pigment. And when that pigment interacts with the iron in the blood, it's going to cause the blood to have this blue pus appearance. And so that's kind of a telltale sign that somebody with a burn has a Pseudomonas aeruginosa infection. Pseudomonas will interface with the air, meaning it needs to be where O2 is present. If I look at where it is growing in a thiobroth, so if this is a thiobroth, where you're going to see it grow is you're going to see it grow in the aerobic portion. So it would grow where the residuant is pink. It will not grow in the anaerobic portion. It's a strict or obligate aerobe. And so this would be our Pseudomonas aeruginosa. Next, we have our Staphylococcus aureus. This is a gram-positive coccus. So notice Staphylococcus, grape-like clusters of round or spherical bacteria. So this is a gram-positive coccus. Did it grow aerobically? Yes. Did it grow anaerobically? Yes. We call this oxygen requirement. This is a facultative anaerobe. Facultative anaerobe grows with or without oxygen, but grows better with O2. So it's going to grow better with O2. So it grew under both conditions though. Now remember that the reason that it grows better when oxygen is present is because when it's aerobic, when it does aerobic respiration, the ATP yield is much higher, about 30 to 32 ATP per glucose, compared to anaerobic. When they do fermentation, they will only get 2 ATP per glucose. So an organism that's a facultative anaerobe will grow better aerobically than they will anaerobically. In terms of diseases that are associated with Staph aureus, we have various skin infections, skin boils, abscesses, necrotizing fasciitis, which is flesh-eating disease, sepsis, so blood-borne disease. It's actually a common cause of food poisoning. Most people don't think of food poisoning as being caused by Staph aureus, but it can be. Staph aureus can also be the cause of pneumonia. And so those are some of the diseases that are associated with Staph aureus. We will learn a lot more about Staph aureus in our last unit in lab. Staph aureus produces this staphyloxanthin pigment. It causes the bacteria to have that golden yellow color. Remember the aureus, the AU is for the atomic symbol for gold. And so that's because Staph aureus, when it grows, it produces this kind of golden yellow pigment. And so this bacteria does not have flagella, so no flagella for motility. So notice that when we see this grow, here you can see it growing aerobically. So it's growing where the rhizagirin is pink. And notice that in the anaerobic, it grows, but this is going to be a semi-solid. And so because it does not have motility, it only grows along the stab line. You're going to see in a minute a different example of a facultative anaerobe, and you'll see E. coli, for example. E. coli is going to basically grow more throughout the bottom, meaning all throughout, but that's because E. coli is motile. It has flagella, and it can move through the agar. In the case of Staph aureus, it still grows aerobic and anaerobic, but notice that it looks a little bit different anaerobically simply because of its lack of motility. Next, we come to Clostridium sporogenes. This is a gram-positive rod. So notice we've had a gram-negative rod, we've had a gram-positive coccus. Now we have a gram-positive rod, or bacillus-shaped. Did it grow aerobically? Right? Did it grow aerobically? No. Did it grow anaerobically? Yes. So we call this a strict or obligate anaerobe. So strict or obligate anaerobe. It only grows anaerobically. And the reason that it only grows anaerobically is because it lacks the superoxide dismutase. It lacks the catalase. And as a result, by not having catalase, they can't basically neutralize those toxic reactive oxygen species. And so for them, oxygen is toxic. So they can only grow in the anaerobic portion. Now, Clostridium sporogenes itself does not cause disease, but there are other Clostridium species that do cause disease, like botulism, tetanus, gangrene, colitis. Remember the inflammation of the colon that's caused by Clostridium difficile, C. diff. So Clostridium sporogenes is an example of an organism that is a nitrate reducer. You guys will learn later on about nitrate reduction. It doesn't interface with air. It does have flagella and it does have some motility. If we look at the way that it grows in the thiobroth, it does not grow in the pink portion. So it does not grow in the anaerobic, or the aerobic, does not grow in the aerobic. It only grows in the anaerobic portion. And so notice that you see it only underneath the pink. So this would be our obligate anaerobe. Next, we have Neisseria meningitidis or Neisseria gonorrhoeae. This is going to be a gram-negative coccus. And in this case, this organism is what we call a microaerophile. Microaerophile is a little air lover. It requires a low concentration of oxygen. So not full oxygen, not fully anaerobically. It requires just the right amount of oxygen. So if we're talking about a microaerophile, can we grow it in our anaerobic jar experiment? Would that ever show us what we would need to see? And the answer is no. It wouldn't grow because it can't grow aerobically and it can't grow anaerobically. It only grows where oxygen concentrations are low. Now, in terms of the diseases that are caused by those Neisseria bacteria, so again, Neisseria meningitidis is going to cause meningitis, which is basically an inflammation or an infection in the meninges, so in the nervous system. And so meningitis is basically transmitted through sharing of saliva. So kissing, for example, sharing drinks, coughing can even happen, etc. And so Neisseria meningitidis can cause meningitis. Neisseria gonorrhoeae can cause gonorrhea. So this is a sexually transmitted infection. What used to be easily treatable, meaning Neisseria gonorrhoeae used to be fairly easy to treat with antibiotics, has now become a lot more difficult because now we're starting to see more strains of multi-drug resistant gonorrhea, meaning that this bacteria has acquired the ability to become resistant to a variety of antibiotics that we would typically use. And so it's becoming more and more difficult basically to treat it because bacteria have become resistant. If we look at a thiobroth to see where it grows, notice it does not grow in the pink. It's not down here in the anaerobic. It's right at the interface of where the pink and the clear meet, meaning it's right where the oxygen concentration is low. It's not too low. It's not down here. It's not too high, meaning it's not in full oxygen. It only grows right at the interface of where the pink and the colorless meet. It grows where oxygen concentrations are low. So this is just to show you some examples of these thiobroths. So we have our Staph aureus, which notice this one has a kind of funky appearance, but basically you're seeing that it grows in the anaerobic. It also grows in the aerobic. Same thing if I look at E. coli. Grows aerobically. Notice both of these grow better aerobically than they do anaerobically. Both of these organisms are an example of a facultative anaerobe. Grow with or without oxygen, but better with oxygen. So we call these a facultative anaerobe. On the right here, this is just an uninoculated thiobroth, basically showing you that the bottom has no growth and the top has no growth, but it's pink because that's where the oxygen is present. And then lastly, we have our last set of organisms. So we have Bacillus megaterium, and Bacillus megaterium, notice, is only growing aerobically. It's not growing anaerobically. So that is our strict or obligate aerobe. If we look at Clostridium sporogenes, notice that it's only growing on the bottom portion and it's not growing where it's pink. This organism is a strict or obligate anaerobe. It's only growing in the anaerobic portion. And then lastly, we have our Neisseria sicca, which is just another species of Neisseria. This, remember, is going to be a microaerophile. And our microaerophile is going to grow where oxygen concentrations are low. So it doesn't grow fully aerobic. It does not grow fully anaerobic. It only can grow where oxygen concentrations are low. And so we see this little band underneath where the bacteria or where oxygen would be found. And so these are just different examples of thioglycolate medium or thiobroths and where bacteria would grow. And based on looking at these, you should be able to tell me, is that organism an obligate aerobe? Is it an obligate anaerobe? Is it a microaerophile? Is it a facultative anaerobe? Based on looking at a thiobroth. And so you would just need to look at where growth is. Remember that growth is going to show turbidity. Turbidity is an indication of growth. So where we see cloudiness is where the bacteria grew. And so this concludes our aerobic and our anaerobic jar and our thioglycolate media.